Hello, everyone, and thank you for joining today's webinar on the development of biomedical knowledge bases for Exposomics. This is the 12th webinar in the Exposomics webinar series from the Human Health Exposure Analysis Resource Program. My name is Chris Duncan, and I'm a program administrator at the National Institute of Environmental Health Sciences. It's my pleasure to introduce our speakers for today, Dr. Carolyn Mattingly and Dr. Anthony Williams, who will be focusing on how knowledge bases can be utilized for advancing exposomics research. Accessible and well-maintained knowledge bases are critical components of a modern data ecosystem, and they play a key role in data and knowledge discovery, integration, and data reuse, as outlined in the FAIR principles. Biomedical and chemical knowledge bases are essential resources in exposomics as we strive to better understand the links between environmental exposures and health. Today's talks will highlight knowledge bases such as the Comparative Toxicogenomics database and the EPA Computational Toxicology Chemicals Dashboard, as well as other web-based software applications that can advance exposomics research. So great talks today. Both of our speakers welcome your questions today. So if you do have questions, please feel free to use the question and answer function on Zoom, and we'll do our best to address each of these questions after the talks. And then we do have an option as well to raise your hand and ask a question, so we can have time for that as well. So with that in mind, let's move along to the presentations. Our first speaker is Dr. Carolyn Mattingly. Dr. Mattingly is a distinguished professor and head of the Department of Biological Sciences at NC State. She earned a BA in art history from Oberlin College and a PhD in molecular toxicology from Tulane University. Her research program aims to improve understanding about the effects of environmental exposures on human development and health. She has led development and expansion of the publicly available Comparative Toxicogenomics database since 2001. This resource provides curated data on the complex connections between chemicals, genes and proteins, phenotypes, diseases, and exposure information. She's also the principal investigator and director of NC State's Superfund Research Program Center, the Center for Environmental and Health Effects of PFAS, which aims to understand about the toxicity, bioaccumulation, remediation of this class of global contaminants. Today, Dr. Mattingly will be speaking about the Comparative Toxicogenomics database, or CTD, and she'll be sharing insights on how this database can help us advance understanding on how environmental exposures affect human health. Welcome, Carolyn. You have the floor.
Great. Thanks, Chris. And thank you, everybody, for being here and for the invitation. So I will go ahead and try to keep this to a tight 20 minutes or so. Okay, so I'm going to give you a little bit of background just about the resource in general, and then focus a little more on some of the exposure-specific information. So the Comparative Toxicogenomics database, it's a publicly available resource that really tries to address the challenge that most chronic diseases involve genetic factors and environmental influences. So we started developing this, as Chris mentioned, way back in 2001, and it's evolved quite a bit since then. So I'm going to show you a little bit about how it's structured and the data that's in it. So I think this is an audience that's familiar with the concept of genotype interactions with the environment and how that's going to result in phenotypes or diseases. And the environment can clearly be defined in very broad ways, from environmental and occupational exposures to lifestyle factors. And for the purposes of CTD, we have a little narrower of a focus in the sense that we are looking at chemicals that are in the environment, and they can, of course, come in lots of different forms and exposure routes. So CTD, the goal of this database as a whole is to help understand how environmental exposures affect human health. And this schematic shows kind of a very high-level view of the overall content that's in the database. And essentially, the information that we're providing to users is a combination of manually curated information that we pull from the peer-reviewed literature. And we focus primarily on chemical, gene, phenotype, disease, and exposure information. And I'll show you more about what that looks like. So we go into the peer-reviewed literature, and we're pulling out very specific types of information and integrating that in such a way that you can, as a user, make connections that might not otherwise be obvious if you're just, kind of looking through the literature on your own. So for many years, we curated the information that's shown here. We consider this sort of our core curation, and it consists of chemical gene interactions, chemical disease relationships, gene disease relationships, and then chemical phenotype relationships. And by phenotype, we mean sort of biological responses that are not exactly diseases. So it could be at a cellular or a tissue level, but that could be valuable in the sense that it helps understand the progression towards disease, or may provide insight into biomarkers of diseases. All of this information is captured using control vocabularies, so that helps us ensure quality control, consistency of our curation, and then interoperability with lots of other resources that are using similar types of vocabularies and ontologies. So just another high-level view so you can have a sense of what kind of data coverage we have. So we have curated content for about 17,000 chemicals, 42,000 genes. That's because these data are captured across many different types of organisms. And in some cases, it can be a little hard to align orthologous relationships across different species. So once we start integrating the data, you can see towards the bottom there, chemical gene interactions and associations that we can build between, by virtue of integration of these data, the numbers get pretty big, pretty fast. So, okay. So what this looks like from a curator point of view, this is kind of a quick view of an abstract. So we do triage the PubMed literature. Our curators initially look at abstracts, but most of the time they're actually going to the full text to capture the full breadth of information in a published piece of work. And what's shown on the right-hand side in the rectangles are examples of pieces of information that are pulled out in a standardized manner. And then at the bottom, these are just examples of kind of the structured format. So we have chemicals that are related to particular genes in this case. And when I say genes, I'm also talking about proteins and the full gamut of the form in which a gene may have been influenced by an exposure. So it could be the promoter region is activated. It could be protein levels are increased. So there's quite a range of different types of responses to exposures that we capture. So we capture this information, as I said, from the scientific literature that's manually curated with the help of some text mining and triage tools that we've developed. We also import a number of other public data sets, so things like reactome pathways, gene ontology information, to kind of fill in some other gaps and not be redundant with other existing resources. And then the power really comes in the integration of this information. A lot of the papers are looking at binary relationships, like this chemical we're showing interacts with this particular gene, and maybe some other paper is then showing that that particular gene has some relationship to a disease. Those connections between the chemical and the disease may not be apparent to somebody coming at these questions from different perspectives. And so by virtue of being able to pull all this information together and make these connections in CTD, we can try to help the users make connections then across these different data types. So our homepage wouldn't be a database talk without a couple screenshots. So just to orient you, this is what you would look at if you were first coming to CTD. There are a number of different ways to access data. You can go through this top navigation bar. We have a number of complex, more advanced query forms. You can search by clicking on any of the entities in our little radial picture. And then by and large, most people come to the database through a keyword search. So you can come at the information from a chemical perspective, a gene perspective, a disease perspective, and so on. So if you were interested in a particular chemical like PFAS, for example, you could do a keyword search and see categories of data that are in the database. And in our case, if we went to the chemical page, you would see something like this. So we have pages like this for from a gene, disease perspective, chemical perspective, and it kind of orients you through this sub-navigation bar where it says basics, gene interactions, etc. All of the information that we have curated and otherwise for this particular chemical. And so this is where it starts becoming, I think, very relevant to this audience. Oops, sorry, let's see. So there's the, yeah, I forgot I had that. Okay, so if we were to go to a gene interactions page, what you would see here are all of the genes for which we have curated information with PFAS and a translated kind of readable sentence that's derived from our curated content. So you can click on any of these factors. So for example, I mentioned we have corollary pages for genes. So everything is very, very tightly integrated. If you're interested in diseases associated with a particular chemical, you could select the disease page for that. And I'll give you a little rundown of what goes on here. So for PFAS, you can get a high-level view of all of the diseases that have been associated with this particular chemical. And we can show you both curated associations as well as inferred associations. So some of these have maybe more direct evidence behind them than others. If you were interested in a particular disease outcomes, such as diabetes shown here, there are a couple of different ways of interpreting this relationship with PFAS. So in this direct evidence column, if there is an M, which stands for mechanistic, or a T for therapeutic, it means there's actually evidence in the literature to support that relationship. But in addition to that, we also have this set of genes, and these are inferred relationships. So as I mentioned earlier, we have PFAS and a disease connection, but how does that actually happen? And in a lot of cases with exposure papers, there may be a correlation between a chemical exposure and a disease, but not a lot of information about what the underlying mechanism might be. And because we have these inference networks, the user can start to build this information set out. So in this case, there are 115 genes that we have curated relationships with PFAS, and those 115 genes have relationships with diabetes. So it can start to help inform what those underlying mechanisms might be. Similarly, we have some tools that are built in here that can give you some more information about these 115 genes. You can look at an enriched disease set. So in addition to diabetes, are there other diseases that seem to have specific connections with these genes? And does that make sense in the context of diabetes? So things like metabolic, diseases, nutritional and metabolic diseases. Here, you can do a Go enrichment analysis, and this can give you some insight into the collective functions of these genes. We have pathway analysis here, and then we also have some additional information with some tools by which a user could say, okay, if among these 115 genes, what do we know about how they interact with each other? So we have lots of tools where you can also input your own sets of genes. Say you've done some sort of RNA-seq and you have a set of genes and you want to know something about chemicals that are known to interact with those genes. You can do something like a set analyzer. Our most recent tool that we've added is called the tetramer tool, and I'll show you a little bit about what that looks like. So this is a little bit of a nod to adverse outcome pathways, the idea of making connections from a chemical exposure to a disease via all of the in-between steps, for lack of a better way of explaining it. So what kind of genes are being affected? What kind of phenotypes? So in order to create a tetramer, we require 5 lines of supporting evidence. So a chemical has to interact with a gene, the chemical has to interact with the phenotype, the disease. We have gene disease connections and then gene phenotype connections. And then we can compile all of this together and say, this is what this collective connectivity might be between a chemical and a disease. So this can get pretty complicated. And we have tools now that will help build these complex networks together for the user. And just for examples, we've had a couple papers recently that have walked through a number of examples. So on the left, ozone and myocardial infarction, and on the right, PFAS and asthma. And if you're interested, you can check out these papers and see how PFAS or CTD is helping to build these networks. So a few years ago, actually more than a few years ago, we started looking at a lot of the exposure data that was coming out and felt like there was additional information that we could be adding to CTD to support exposure science. And so for this group, I don't have to explain what the exposome is. We convened several exposure scientists and people in the field, and we're trying to get a handle on what exactly the information was that would be useful. And it was clear that the structure, we needed to structure the curation around something that didn't quite exist yet. And it was quite complex in terms of the data that needed to be captured. And so we built an exposure ontology, and this stressor, receptor, exposure event, and outcome is kind of the framework around which we have built this curation. So what this looks like from a very high level is our stressors, in our case, are chemicals and source information. Our receptors you can think of as the individuals who are exposed to a particular exposure scenario. The exposure event is kind of what's measured in the community or in households where this happened. And then the exposure outcome can be any number of phenotypes or diseases. And this looks very simple. It's actually quite a bit more complex than our core curation that I started with here. It can be very labor intensive for our curators. But I'll show you what it looks like at the end of the day. And the important point here too is that the content in gray represents data we were already using in CTD. And so a lot of the exposure curation, the details are new, but the content is highly integrated with the existing structure of CTD. So our stressors are connected to our chemicals, exposure events are connected to our chemicals and genes, and then of course outcomes are connected to phenotypes and diseases. So if you're in CTD, again, we're back on a PFAS example here, and we're just navigating further down the options of curated data. So if you go to an exposure study tab, what you'll see is like a high-level view of all of the exposure studies that we've curated for PFAS, as well as whatever other chemicals were studied in that particular paper. So each row here, in this case, we have 193 studies, exposure studies that were looking at PFAS exposure in populations. And then you can see this sort of is in line with that framework that I showed you in the previous page. So stressors these are all the chemicals that were studied in each of these publications, the receptors. You can see a high-level description of what the population was for that particular study, and then what was measured and in where. So this is really a summary view of a particular paper. And then if you want more details, like what were the actual levels that were studied, you can click on the details, and what you'd see is this. And it's probably a little bit hard to read, so you'll have to take my word for it or go to the database and see for yourself. We have about 50 different types of information that our curators are capturing. Our default is about 12 of those, just for space sake. But as a user, you can toggle on and off any of these categories of data and determine what you want to see. And what we're looking at here is an exposure detail page. So this is a single study and all of the information that was reported in that study. And so it can be very detailed. So these are different subsets of the populations. There are notes from the study. There's information about the sex, race, age, if it's reported. You can see here that different chemicals were captured using serum in this case, and then what the particular levels were that were measured. And then if there were any outcomes that were positively or negatively correlated with exposure. So again, you can access the exposure study information directly from any of the high-level categories of data we have. We also have advanced search options if you want to go in and be very specific about, rather than just coming at it from a chemical perspective. For example, you can look at all of the NHANES data. You can look at the information by study if you want. Lots of options there. And then of course we have all of our data, including the exposure data, can be downloaded anytime you want from our download page. So lots of ways to access this information. And then again, just to wrap up, the nice thing about the exposure information, you can, and what I'm referring to as exposure information are really like epidemiological studies where people have gone in and measured chemicals in households, for instance. That information can be separate, but it's also integrated with the core information. So for example, if you're looking at an exposure study in the context of a particular chemical, that chemical also has associated with it all of the other information that I started with. So gene, phenotype, mechanistic type data that's derived from very different types of studies. So you can start to think about the data. The exposure scientists are doing the real hard part of, going in and measuring in populations and homes and occupational settings, and may not be thinking of or setting up those studies in the context of mechanistic understanding, but we're trying to do that for you. So that information is all accessible. So I'll just end with an overview of what we're providing from CTD. Again, the curated content is coming from the literature. We're manually curating that into behind the scenes, these kind of binary data sets, and then pulling them into more complicated, complex inferences that can really be used to support a systems toxicology approach. So if you've got questions for any of us on the team, you have curation requests, you need help with more complex queries that our query forms may not directly support, just contact us. There are links to do that on every single page on the database, and we're always happy to hear from folks and help you navigate the database. And we have a fantastic team, many of whom have been with the database for quite a long time, so very dedicated and creative group, and we're very grateful to our continuous funding from NIEHS, and particularly the U24 mechanism, which has been extremely helpful to us in the last few years. So with that, I will stop, and I'm happy to take any questions.
Thank you so much, Carolyn. It's always great to hear about the amazing work the CTD team is doing. So thank you for presenting. We do have time for a question or two. I'm not seeing any in the chat right now, but please feel free to add your questions to the Q&A box. I might start off with a question. So could you comment on how you prioritize studies, particularly the exposure studies, into that exposure module? How do you prioritize the many important studies that are being curated?
Yeah, that's a great question. Obviously, the manual curation process can be a little bit slow. So we do have a couple different ways we do that. We have sort of what we call the currency approach. So we have a number of journals that are relevant to toxicology, or high profile journals that we prioritize. So we go through those on a monthly basis and pull out papers that are relevant to CTD, in the sense that they have to have some sort of chemical and other content that is included in CTD. So there's that approach. And then we also have chemicals and classes of chemicals that are of priority. And we base that on NIEHS priorities, things that ATSDR are looking at, EPA. So we try to hit a lot of the chemical classes that are, of particular relevance to those communities. And then, of course, we get requests occasionally from users who are looking at a chemical that maybe hasn't hit our priority list, but it would be of particular value to their research program, and we try to accommodate that as well. So yeah, the exposure space, the landscape of those publications relative to like our core curation, sort of more molecular kind of work, there's definitely not as many papers there. That's not to say there aren't, but that's also changing. So when we started this project, it was like a couple thousand, and it's definitely changed since then. So we do our best to try to stay on top of it, but it'll be tough to get everything.
Yes. Thank you so much, Caroline. We do have one question in the Q&A box. So what was the p-value in your search results? For example, in your diabetes examples with PFAS, there's many values. What do those represent? It's a question from Emma.
Yeah, so that, so I'm looking, so what we'll do is we have a structure by which, so we're basically working off of what the authors have presented. So if there's, so I'm looking at in the assay notes, that would be specific to notes that the authors would have provided in the context of a particular study. So in that example that I showed you, there are many, many lines. So there are 400 lines of data in that particular paper. So in each relationship, the authors may say, well, we found, this level of PFAS in serum from this subset, and that was considered a significant difference, for example, from something else that was measured. So those assay notes are intended to provide a little more context than that the authors may have provided in the publication.
Great.
I hope that helps.
Thank you, Carolyn.
Yeah.
And Emma said thank you as well.
You're welcome.
So I think in the interest of time, let's go ahead and go to our second speaker. Our next speaker is Dr. Anthony Williams. Dr. Williams joined the Center for Computational Toxicology and Exposure and the Office of Research and Development at the US EPA in May of 2015. He's a chem informatician focused on the delivery of his center's data to the scientific community. His interests include the aggregation and curation of chemical data, development of models to support physiochemical property prediction, and development of software approaches to support non-targeted analysis. He has over 2 decades of experience in chem informatics and chemical information management, with focus on internet-based projects to deliver free access community-based chemistry websites. Example here, he's the founder of the ChemSpider database, which provides access to data for over 115 million chemical substances. And he was also the product owner for the EPA's CompTox chemical dashboard for 6 years. And now he's leading development of 10 proof of concept software applications, including hazard profiling of chemicals and structure searching, and also an open spectral database, as well as a corresponding analytical methods database to support non-targeted analysis. Today, Dr. Williams will be speaking on utilizing US EPA data dashboards to support exposomics research. Welcome, Anthony, and take it away.
Thank you. Can I confirm you can see my slides?
We can. They look great. Thank you.
Excellent. The US EPA, and I'm at the Center for Computational Toxicology and Exposure. Let me just switch a couple of these screens down. Okay. One of our primary roles is to develop curated chemistry data streams. And a lot of our tools that we release out to the public sit on top of these this chemistry database. It's been in development for over 20 years. It's only been serving the publicly facing applications for about 8 years. I'll tell you a little bit more about that as we go. The prediction models, web-based applications that we build on top of this is to support many, many different applications. I will touch on some of them today. I won't have time to do all of them for sure. There are so many different methods and applications that we develop on top of these data. And I want to focus today specifically on what do we do to support exposomics and especially some of our efforts to support non-targeted analysis around mass spectrometry. So DSS Tox database was originally started by one of my colleagues who's now retired. Her name is Dr. Ann Richard. When she started, it took a number of years just to get up to 5,000 chemicals. I think it was about 3 years of work. Today, of course, things have improved a lot about how data can be assembled, how we can we built an application for curating these data. We have full-time curators, much like Carolyn was talking about. We have very specialized skills for curating our chemistry data. We build relationships between these chemical substances. So, for example, a parent chemical and its disinfectant byproduct, parent chemical and its transformation products in human, et cetera, et cetera. So we do a lot of work to map these relationships. All of the data mappings are done under this underlying database called DSS Tox, and you as the community get to access it via the dashboard. And the dashboard, the CompTox chemicals dashboard, we refer to it affectionately as just the dashboard, has been available for a number of years. I've been here since May the 1st, 2015. We started work in December of 2015. We made it public in 2016. So it's been online for a while now. It is now up to 1.2 million chemicals, lots of related data. So experimental and predicted phys-chem data, experimental human and ecological hazard data. We've got ToxCast, Tox21. I'm sure many of you have heard about the bioactivity screening data that we have been working on for well over a decade. We now integrate data to consumer product details, actually down to the level of the product name itself. We can give you information on chemical weight fraction. That has just come out as a new database. I won't touch on that in any detail today, but if you're interested in that new database, please do a search. We also link to other agency websites and public data resources, and we do a lot to support a lot of that mapping information I'm telling you. And while many people are happy to look for a single chemical at a time, just as Carolyn was mentioning, look for PFAS, she has a batch search. Batch searching is essential for what we do. People are not looking for one chemical in general. They're looking for a set of pesticides, a set of PFAS chemicals, et cetera, et cetera. So batch searching is an essential part of what we do. I'm going to take the example of perfluoro-related chemicals also. If we could search here for PFAS, but I just want to find the set of perfluorochemicals. So I can just start doing a type ahead search right here, and you will see the list. If you type in BPA, you will get the CAS number, the name for BPA, as is shown here. So if that, in this case, if I search for BPA by CAS number 80057, you can see it at the top. BPA, any of its synonyms that we have mapped, I think we have 80 synonyms for BPA mapped. It will bring you to this record. And under this record, you see on the left-hand side a lot of navigation details into properties, hazard data, exposure data, what I was summarizing at the beginning. So 1.2 million chemicals you can navigate into. Not every chemical has this richness of data. So not all the time are we going to have experimental hazard data for sure. We'll always have synonyms. We don't even have to have CAS numbers all the time. Not every chemical that we register has yet shown up in SciFinder. So this database is continually maintained, full-time curators expanding every day and updated to the community on a fairly irregular basis, at this point about once a year. So we have the search entry point, we have the tox data. Every time you see a table of data, you can download it into an Excel file. Everything we do is to give away to the community. This is a navigation panel into Tox21 and ToxCast data. We have basic chem informatics mapped in here. So if you search for a chemical, you can find all of the similar chemicals based on a simple chem informatics Tanimoto search. We have a read across module called generalized read across, originally built into the application, but recently split out as its own separate application. You can still access it directly from the dashboard, but it does run independently now. And this allows you to do read across for a chemical using lookup based on Tanimoto search, availability of bioactivity data, et cetera, et cetera. So that's called Genera, G-E-N-R-A. We've also got integration to literature searching. So we've built an application that uses the PubMed API so that you can come in, land on a particular chemical, and then search across PubMed and filter through the data based on specific queries that you might have interest in. And here's an interface to the batch search. So the batch search is incredibly enabling. And I think this is a point which will start to show you really how fast you could harvest data from our application. So you can see on the left-hand side, you have different identifiers that you could put in. Chemical name, CAS number, INCHI key. I'm assuming most of you know what INCHI keys are. DTX substance identifiers. Those are our unique identifiers for our 1.2 million substances. They're everywhere now. They're on PubChem, they're in ChemSpider, they're all over Wikipedia. So if you want to download our entire database, you can go to our Figshare page, download the entire set of data with synonyms, CAS numbers, names, structures, etc. And then you will have access in your own systems, if you integrate them, to these DTX SIDs. You can also do searches based on formula, exact formula. I say MS Ready formula here, I will tell you what they are, and mass. Formula and mass is specifically to support chemical identification in mass spectrometry and non-target analysis. So what I've done here is I've input the set of DTX SIDs. Now what do I want to download from our database? Because you would like to harvest information such as the SMILE string, the mass, maybe whether or not the safety data are available, what our toxicity data are in our ToxVal database, what information do we have and whether these chemicals are being screened, et cetera, et cetera. Everywhere I have clicked a button in here to get to check the box, that will download immediately. Take it just a few seconds for a few hundred chemicals. It will bring it down right into an Excel spreadsheet. You can go through all of the information we have on toxicity, exposure, et cetera, in an Excel format. However, I would say that while this is useful to you, you can filter through it. There are nicer ways to show these data, and I will highlight a couple as we get towards the end of this talk. Excel data is a great way to share data, not necessarily the best way to visualize it. I'm going to step back for a moment. We have 1.2 million chemicals, but commonly those chemicals have come together under specific efforts. So, for example, we would like to release to you the TSCA inventory of active non-confidential chemicals. So we have built a list that's 36,000 chemicals. It is versioned. We update it every time TSCA updates. So you can download the TSCA inventory of CAS numbers, names, SMILES, et cetera, one click, and you have the entire TSCA inventory. It is subsetted into active, inactive, and non-confidential. We cannot share confidential business information via this application. We will also have a list for the Clean Water Act, has substances. There's a regulation coming out from the EPA by the end of the year. That list is already available. Consumer products database that we built, chemicals of emerging concern, extractables and leachables, 425 lists and growing. This week I've worked on 3 more. They will be updated in the next couple of weeks. Here's an example. So the LC database is a public resource. We access the LC database, we download it, we curate it, we make sure it maps into all of our chemical substance details. We put it up as its own independent list. We link back to the original source to maintain provenance. So you can go source it there. But if you want all of the chemistry data and all of our related properties and toxicity, you do it from our site. When we work on things like thiochrome rubber, so we released the thiochrome rubber characterization report in 2019. We did all of the chemical curation. So you, as well as the PDF file, you can access all of the chemicals here, just in case you wanted to download them, use them in your own databases, build models, understand what's in there in a very facile manner. Same would be true for hydraulic fracturing. Hydrofracking, of course, major concern. So that list also exists. When we have regulations and we release them, we do all of chemistry. Susan Richardson has worked, ex-EPA, she has worked for many years in disinfectant byproducts. So we work with her to make sure that we keep an updated list of disinfection byproducts as we build it out. Going back to PFAS for a moment, we know this is a concern worldwide at this point, of course. So there is a lot of effort going on in the agency to deal with PFAS. We assemble all of the PFAS lists. They can be categorized differently using different substructural moieties for different purposes. So the 8A7, this is the latest PFAS regulation that came out from EPA. The 8A7 list was finished last night. That will be released in the next 7 days. We have all of the drinking water PFAS methods listed in here, the OECD global database. So we are staying on top of PFAS as much as we possibly can and releasing the data out to you. So switching now to measurement data. So we, of course, measure a lot of data ourselves in terms of detection of chemicals out in the environment using mass spectrometry. And we need to know what chemicals are detected in the environment. Ideally, we would like to do quantitative non-targeted analysis. Significant amount of work. We've been working on it for 3 years. We're making great progress. But we want to make sure that these measurement data are available, that we can characterize what is out there in the environment, so we can identify the hazards of these chemicals. We can do some form of assessment, risk characterization, and ideally this will all move forward to support all of our regulatory efforts. So we're doing a lot of work. Non-targeted analysis is a very focused area for us at this moment. We're expanding our team. We're building our own applications to support it. One of the things we've had to do with all of the chemicals in the dashboard, 1.2 million substances, they're of all forms. stereoforms, salt forms, multi-component systems. But in mass spectrometry, you are detecting, using the technique, you're detecting individual moieties. So you won't be so sensitive to stereochemistry. You won't know whether a chemical is a sodium salt, potassium salt, how it found its way into the environment. If you're finding it in water, you won't know what the counter ion was that was associated with it. However, by mapping together all of these complex forms down to a mass spec ready chemical that you would detect, in this case, at the middle of this, we have MS ready form of nicotine, de-stereoed, desalted, single component, that comes from all of the other related substances, which may have CAS numbers, names, etc., consumer product identification. By distilling down to this MS ready form, we are supporting the efforts of the agency to do chemical characterization and identification. When we do this, we take out 1.2 million substances, we distill it down to a set of MS ready forms. That gives another set now. Some of them may be virtual in nature, i.e. It's never been reported and as anything other than a part of a multi-component system. But by mass spec, you would identify each individual. When we end up with these hundreds of thousands of distinct chemicals, we have then been able to predict the positive ion, negative ion mass spectrometry, 3 different energy levels. For those of you who understand mass spec, 10, 20, 40 electron volts, positive ion, negative ion. EI spectra, so we have calculated 7 spectra for each of the chemicals in its MS ready form. So we've predicted them using work that came out of Dave Wishard's lab in Alberta. So we've ended up with hundreds of thousands, actually millions of predicted spectra. These go into a database. We can now search our experimental spectra against predicted. We can bring back the best hit matches, and now we have a set of chemicals that we need to determine which are the best actual candidates for us to focus efforts on. Because if you can detect a chemical, but it has no major hazard issues, then you don't want to focus massive efforts into it. You want to go after those ones which you think might be an issue. So we've applied this approach for identifying unknowns. We have been publishing in this area for a number of years at this point. We have used large groups, and TACT, which is mentioned in this screenshot here, is the EPA non-targeted analysis collaborative trial. We have worked with almost 40 labs. We have distributed samples to them. We've collected data from for all of their instruments to see what they could do to do identification. And we've built our own tools as a comparison to see if we can up the level of performance for this non-targeted analysis approach. So we've been reporting on this for a number of years. We went back into this competition that was started work in 2012. So CASMI, the Critical Assessment of Small Molecule Identification, is a great source of data sets that we could test our performance on. We published on that. We were doing it post factum, so we weren't part of the original competition, but we were able to show that our tools at this point in time were really performing at the level of all of the best performers in the CASMI competition. But we didn't stop here. The reason that we build tools on top of our informatics platform from non-target analysis is detection is not enough. When you have a set of candidates, a set of chemicals that you might want to be concerned with, we now know the structures because we've been able to identify them with this approach. But you want to focus your efforts on those that you think might be most hazardous. So what we've built is another public tool. You may or may not have heard of it. This is one of the Chem Informatics modules, publicly available for over a year, where you can input a set of CAS numbers or names, and you can quickly profile those chemicals. So in this case, I've put in about 500 chemicals. I clicked one button and it has pulled together the data from ToxVal, which is our database of toxicity data, which is separated into oral, inhalation, ecotox, endocrine disruption. And what you see in front of you is a heat map for all of the chemicals that we were able to pull the data. The data in ToxVal were harvested from 40 public databases, it's curated, mapped together, and each one of these individual cells in this heat map, when you click on it, it shows you all of the underlying experimental data that we have been able to pull together. This is separate from the dashboard. It still uses DTXS IDs. It sits on the same database of DSS tox data. It sits on the same database of property data. It's the same data that sits under the dashboard, pivoted into a new visualization. So as I said, Excel files are not the best way to profile all data. We think this is a much friendlier way. It is available for you to play with. To the last part of what we've been doing here. So these ongoing efforts to include NTA in terms of Exposomics, we've got a lot of work going on right now with states on PFAS identification, some breakthrough work happening on applying our tools to this process. We've been looking at pesticides in various matrices, chemicals of emerging concern. Biosolids is, of course, a major concern. We have efforts across all of these, including collaborations with universities, and we've even funded some biosolids research. But some examples of what's been published, I can share these slides with anybody. They will go on to SlideShare right after this presentation. This is an example of us screening chemicals and consumer products. We purchase products, we would grind them up, we would run non-target analysis approaches to see what we could discover. We have looked at recycled consumer products, the chemicals contained in recycled products versus virgin products, rather significant. It's interesting to see what shows up in these recycled products. Oh, this is already published. And some work we did on preeclampsia and chemicals we were able to detect. Again, using the same database, the same tools approach, pulling together mass spectrometry data and using our non-targeted analysis pipelines to identify chemicals in totally different studies. So in conclusion, all of our resources we're building in chemistry underpin all of our chemistry related research for the center. We also share it, of course, across the agency. We work with all of the other databases. We do a lot of curation for them. And curation and data quality is key. It's not hard to find a large database. Those are easy to find. It's hard to find a high quality curated database. And Carolyn spoke to that wonderfully. Her work is so wonderful. Our web-based applications to deliver the data for you to use, and there's multiple use cases. The support for Exposomics, it includes the chemistry data streams that we have been building, the experimental and predictive properties, the toxicity, all of that profiling that we can do now is all there for you to use. It's all publicly available. We have other tools in development at present. The NTA web app, non-targeted analysis web app, is going to use all of these data streams. It's going to be made available for you as a community to plug your experimental data into, and it will go through our pipeline and report back to you our identification of chemicals and how we got there, including some very detailed statistics. I get to give the talks, but there's so many dozens of scientists behind the work. We have a very small but dedicated team of curators, the dashboard development team, and all of our software group. I have a number of student service contractors who build the applications for us. Our mass spec team doing some wonderful Exposomics non-targeted work, and all of our student service contractors and postdocs. You're seeing the work of a couple of hundred people over the years. I will make these slides available at this URL. If you want to grab a screenshot of that, slideshare.net, Anthony Williams, I will load them there immediately after this talk. If you want to follow our work, fastest way would be either to follow me on LinkedIn or to go to my Google Scholar profile, and you'll see all of the publications that we put out. I will stop there.
Thank you so much, Anthony. What an amazing collection of data resources and tools. Truly a rich resource. So thank you for your presentation. So we've got a few minutes left, not too many minutes, but with the remainder of the time today, we're going to take some questions pertaining to Anthony or Carolyn's talk, if there's any remaining questions. Okay, feel free to add them to the Q&A box, raise your hand. I think Philip had his hand raised. So Philip, if you want to ask a question, please raise your hand. If not, I can, we'll take moderators privileges. So Anthony, you mentioned your measurement data. So could you comment on what are the sources of those data? Are they coming all from EPA? Is this from external resources? Yes
to all. Yes, we have many collaborators. Sometimes we are not measuring the data ourselves. We get the data from some of our groups like Johns Hopkins University we're working with at the moment. We work with UNC Chapel Hill. We have state data coming in now directly. So it's our own as well as across the nation.
That's amazing. Thank you. Okay, let's see. So I guess maybe a question for both of you. Carolyn, you mentioned controlled vocabularies, EXO, and Anthony, you mentioned your chemical identifiers. So I'm wondering, for our Exposomics researchers and the data producers from our HERE labs and other NIHS supported labs, is there a way that investigators can report their findings that are either interoperable with your databases, easier to ingest into CTD, for instance? Are there particular standards, terminologies that you see as critical to foster interoperability?
I'll let Carolyn go first. I think the chemistry side is easy relative to what she does.
So the short answer is yes, but of course it's more complicated than that, because it's not like every study is structured in the same way. But I think certainly community agreement on which vocabularies or ontologies are to be used when reporting out information, such as genes and chemicals and diseases. There's a little less variability there, but and I think we've come a long way. It's gotten a lot better than it used to be, in part through efforts by NIEHS, I think, to build out awareness of the consequences of not using these structures. So yeah, I mean, I think there should probably be agreement on that. What we use is we use MESH for our chemicals. We use the NCBI genes for our genes. And there's a little debate in the community, I think, over the chemical vocabulary and disease. I think most people are pretty aligned on genes. So it's definitely gotten better, but people should be consulting, I think, with the broader community, and I think that's something NIEHS is trying to take a lead on, in terms of, how this broader community is reporting their data.
From the chemistry side, I would say that if every publication did a simple thing and added supplementary info in a form of an Excel spreadsheet instead of a PDF file that included CAS numbers that have passed checksum, associated name, smiles at ideal, and INSHI key. We now have 4 identifiers to try and curate those data. 1 is never enough. People will put the CAS number for the neutral, but they've actually studied a salt. They will put a non-standard INSHI in. They will have a non-canonicalized smiles. Give us a set of identifiers in an easy to harvest format, and we have tools that will pull that out very easily and map it all back to the publication. So we're doing that type of thing now for analytical methods. So literally the simple issue of spreadsheets rather than PDF files with 4 chemical identifiers makes things very easy. There was a good publication by Emma Szymanski and Evan Bolton about verifying chemistry data. That would help. And then we have efforts going on the non-targeted side. There is a group called BP4NTA. BP4NTA, which means best practices for non-targeted analysis. There is a standard reporting template discussion. Go to non-targetedanalysis.org, and we are encouraging people to adopt these best practices. It makes everything easier for us to deal with on our side, and it makes it richer for the community to get to these data.
Yeah, I would agree with that, because we have there are some, sometimes the supplemental data will come in forms of spreadsheet, and that greatly facilitates the manual curation process. But I would agree with Anthony that having multiple, because some of those chemical identifiers are not even unique. And so, having multiple ways to kind of cross-check what you're actually talking about would be really helpful.
Great, thank you so much. A question from Emma, does the CompTox dashboard have information on disease or epidemiological outcomes related to chemicals? Or is this toxicity data related to more acute endpoints in animals?
So the ToxFile database is acute, chronic, subchronic, et cetera. So you can dive down directly and you can see all types of endpoints. So we've got points of departure, exposure levels, MRLs, NOELs, LELs, et cetera. So there's a lot of complexity to that data. Easy to harvest, but way more than just the acute endpoints. The same data, of course, shows up in that hazard profile I mentioned. In terms of disease and epidemiological outcomes, we have mappings out to systems that will help you with that. Some of the in vitro screening will be suggestive of potential outcomes that you might want to concern yourselves with. We link directly to Carolyn's work via our external links. There's an external links page that links the dashboard out to 70 different systems online. One click and you're into those systems. And I would encourage everybody to take a look at CTD base. It's a phenomenal resource. We do not mean to reproduce that. That work is so well done. We do integrate to the AOP wiki, things like that. So that would help. And then I see there's another question regarding informatics approach to determine chemical of origin and metabolic products. There's a couple of papers that have gone out recently showing how we use our approach with non-targeted analysis to look at metabolites, mapping back to parent chemicals. There's a paper that just got submitted last week that will show our similar approach validating against predicted metabolites and transformation products. There are many tools for predicting metabolites. Our non-targeted approach with these predictive approaches for metabolic breakdown products, we think that is the path to go ultimately. There's a tool from Wishart's group called Biotransformer, which is excellent, but we are testing many to do that. And I think that will be the future of global non-targeted mass spec. We'll be using similar integrated approaches.
Great. Thank you. And I think we are out of time, but in closing, I just wanted to thank both of our speakers for your excellent presentations, thoughtful discussions today. And a big thank you to the HERE Network and the organizers of this webinar series as well for the opportunity to highlight these very important knowledge base resources. So before we sign off, just a quick announcement. The next webinar in the HERE webinar series for Exposomics is going to be in January of next year. And the topic's going to be on advances in multi-analyte quantitative assays. So it should be a very good webinar in January. The specific speakers and date are still to be determined, but this will be released hopefully soon through the HERE program website. And for any more information on that, please go to hereprogram.org. You can find the recordings of this webinar series there, so that's a great resource as well. So with that, just like to say thank you again to our speakers and thank you to everyone who participated today. We'll see you in January. Thank you.
Thank you.